related to enzymes, and the statement says the two enzymes P and Q are proteins with quaternary structures which catalyze the same reaction, but when sorry, but they have different amino acid sequence, mean they are different, two different proteins. Define the quaternary structure. I guess this is easy one for you. What is meant by the quaternary structure? There is more than one polypeptide chain. Uh, what type of bond is present in this structure? Yeah, in quaternary structure, what type of bond is like a sulfide linkage or a hydrogen bond? Oh, okay. So you can mention that these bonds can put these polypeptide chains or connect these polypeptide chains. Okay. You can also say this. So explain how the enzymes with different amino acid sequence can catalyze the same reaction. Like it can happen that a single substrate can can be attached to many they, so they have a similar active site shape. Okay. So if they have a similar active site shape, what is going to happen next? So it makes more enzyme substrate complexes and the product is formed. Okay. This one is quite simple one. Next, a graph is given. So scientists investigated the effect of pH 8.4 and 7.5 on the activities of the enzyme P and Q. So a figure shows the values. So what do you get from these values? We have 8.4 as well and 7.5. Do you find any difference um, or what about their like the which enzyme is performing best at 8.4 sorry, and which enzyme is performing best at 7.5? Is the okay, the enzyme Q you're saying is performing the best. Let's forget the control. Just tell me the difference between this one is Q and the solid, the darker light is P. So you are saying that at 8.4 Q is performing at its best, or it's is showing maximum activity because the concentration of substrate is decreasing. That's how the reaction occur. So if the concentration of substrate decrease, it means the concentration of product is increasing. Okay, what about the 7.5 pH? Both are not performing better. Like they're almost same. They're almost same. So look at this. The lines, they're very close to each other. So let's define it again. So here we are saying the P and Q are performing equally good at pH 8.4, while at pH 7.5 P is performing better than Q. Q is performing better. Okay. So let me explain this. How do we say if something is performing better or not? uh, how quickly it changes the substrate into product. That would be quick, right? So we say P is quicker than Q, or P is converting product more efficiently than Q. Okay, so let's move to the question. The question says describe what the scientist should place in the control tubes in this investigation. Okay, first of all, this what should be the control? Like the control is the one which has the maximum availability of all the factors. So what factors are required? Sorry. Yeah, in control. The optimum what? Temperature, because here we are investigating for pH, so the optimum temperature first of all. Okay, what's the next one? No, the pH is changing. It's 8.4. Okay, we can say the constant pH in both, and that pH is maintained by which solution? Buffer. So we'll use a buffer solution. Okay, what could be added? The same concentration of substrate in P and Q. Okay, same concentration of the denatured enzyme. Okay, sorry. Okay, so we have used a buffer solution, we have used the optimum temperature, we have used the same concentration of substrate and the denatured enzyme. Okay, correct. Now, what are the three conclusions that we have discussed from this figure 8, the graph? What are the three conclusions that you can make from this? So first of all, we say at pH 8.4, enzyme P and Q, they are equally good or they are performing at the best. But if we discuss pH 7.5 here, P or Q is less active than P, or P is more active than Q. Okay. What other, because it's of 3 mark, so we need to give the conclusion. Like, what do you think? What makes this activity? What makes the activity of Q less? Maybe Q is denatured due to the change in pH, which is 7.5. So this can be conclusion. Okay, first one we have made at like at pH 8.4, both enzymes P and Q are uh, active. At pH 7.5, P is more active than Q. What can be the reason? Q may be denatured. Okay. Okay, so let's move to the next question. The next question is again a graph is given, and they're asking about the transpiration rate. Okay, let's first of all have a look on graph. So what does this graph is telling? Mangrove trees grow near the sea. Seawater surrounds the lower part of the transpiration. Okay, first of all, this solution would be concentrated as it would have more salt. Scientists investigated the rate of transpiration in mangrove tree. A different, like something is being shown. Okay, first of all, the rate of transpiration is given by a solid line. The rate of transpiration is about 0.39 at 5 a.m. Then around 11 a.m. it is 0.84, let's say, and this one is the maximum one. Then at around 2 o'clock, it's 2 o'clock, it's 0.80. And then at 7 p.m., again, it returns to its original, which is 0.39. Okay? So this is what a graph is telling us. So there is decrease at 1 p.m. They have shown a decrease at 1 p.m. Okay. Now, explain the rate of transpiration between 5 a.m. and the midday shown in figure 9. Now, whenever there is a word explain, we need to keep in mind, we need to give reasons that why is this happening. Okay, in description we just write the result, okay, this one is increasing or decreasing, but in explanation we need to give the reasoning. So what can be the reason of this increase or decrease? Okay, so if the more of the water to leave. Okay. What else can be added? So first of all, you mentioned that the rate of transpiration increase because of the increased temperature. That is correct. That is your first point. Wait. Then you have mentioned light intensity. First one is about temperature. Okay, light intensity. And what reasoning have you given? Okay. Okay. What else can be? It's related to transpiration. So relate something with transpiration. Stomata. Stost. Yes. More exchange or or you can say carbon dioxide and oxygen exchanged, or carbon dioxide enters the leaf for photosynthesis. We can also say this because the stomata are open. Then at 11 a.m., so it's not 11, it's 1. Okay, here where the rate is low, it's 1. Yeah, after 11 a.m., or you can say 1 p.m., the rate is low because the stomata close due to conserve water, to reduce transpiration. Okay, so here they have mentioned 1. Here. Okay, 1 p.m. I thought it's at 1 because it's a solid line, it's going at 1. This one solid line. Okay, I guess my screen is off. Okay, are you sleepy today? Was it a tiring day? And what did you do? You were simply tired. Was there any event? Okay. Okay, let's move to the next question then. Okay. The next question says use figure 9.1 to calculate the percentage increase in the rate of transpiration from 1 p.m. to 2 p.m. Now we need values, so we need to go back to graph to have values. Let's go back to graph. So what is the value at 1 p.m.? It's around 0.75, and at 2 p.m. it's 0.80. Okay, so we say it's 0. It's 1 p.m. Okay, it has increased from zero point 0.5 to 0.8. Okay, let's find the percentage increase. It would be the final value minus the initial value divided by the initial one, multiply by 100. So it would be, I guess, 0.05 divided by 0.75 multiplied by 100. I guess it's 0.05. So what's your answer? You need to calculate the whole thing. 0.05 divided by 0.75 multiplied by 100. 5 divided by 0.75 multiplied by 100. So the answer is 6.6. Okay, or we can also say 6.7% if we round it off. Okay. The next question says the higher rate of transpiration at high tide shows that the tree is absorbing water from the seawater surrounding its roots. Describe an experiment that you could do to investigate whether the mangrove root cells have a lower water potential than seawater. And you have three or four more things. Number one, you have a piece of fresh root, you have that seawater with the same concentration, obviously, and access to the laboratory equipment. So what equipments you are going to use first of all, and how you are going to use that route. Microscope, okay, the equipment would be optical microscope. What else do you need? Would you need slides or anything? Slides. Okay, optical microscopes, then slides. And what to put on slides, and or like how would you like place your sample on that slide? Tissues. Tissues from? Tissues from this root, from plant root. Okay, now first of all you need to describe uh, how you will place these tissues, and how you are going to check the effect of the seawater on the, like, whether the osmosis has taken place or not. Okay, how you will say that? First of all, we need to take the initial, okay, initial mass, okay, initial mass or length, okay, initial mass and length. See water for a given time. Okay, don't say sometime. Write a word given. For a given time. Okay. So we need, okay. One thing. We need to dry it before calculating the initial mass. Okay? Okay, not after. Okay, after we have placed this in seawater, now we have taken it out from the seawater. First, the very first thing that we need to determine is the change in size or mass. Okay, or increase in length. So the increase in size and length shows that osmosis occurs. Now, what is osmosis? So, okay, you have used this optical microscope. Why you have used this? To check something. Cells become wrinkly, swollen. So we have seen in optical microscope that the cell become swollen, and in optical microscope we can easily see the vacuole. If we stain the whole thing, the tissue, if we stain it, we can clearly see the turgid vacuole. Okay. Okay, so let's move to the next question. Just give